Hello, can somebody confirm you can hear me?
I can hear you.
Thank you. Sorry for being a little bit late. I didn't notice the time, and there's 12 things I have to do to get a Zoom session started. The security system has gotten really ridiculous. So before we begin, are there any questions?
I have a question. Are you going to share the slides for chapter 5 and chapter 6?
I haven't done that yet. Yeah, let me try and do that right now if I haven't done it. Where is that? It won't work. Let me share my screen here. You should be seeing my screen. Somebody have your microphone on, just to let you know. That's the lab. Have we finished chapter 6? Let me see where we are. 521. We have. Yeah, if ever I haven't done that after a day, you can send me a reminder. Obviously, chapter 5 has been a while. Let's go back to chapter 4 just to make sure. I do have it for chapter 4. All right. So today is May 21st, and we are supposed to be working well on chapter 8. I think we're only starting chapter 8. We're a little behind schedule. There will be a lab. That doesn't seem right. Oh, sorry, I'm not looking at the right thing. There will be a lab today. It's lab 9. And on Wednesday, tomorrow, you will have a quiz 4, and it will cover chapter 5 and 6 and lab 7. Any questions about any of that? If there's no questions, let's go ahead and get started with chapter 8. Come on. And we're only on slide 7. We should be about halfway through chapter 8. So we had talked about the vertical flow of information within a cell. There is also, besides the flow of information within a cell, there's the flow of information between generations of cells. And for humans, you'd say between generations. Let me blow this up. Where information can flow from a parent cell through replication. Can somebody shut down your microphone?
No, not
Sorry, I have to figure out who's doing that. There, I've muted Leela. Okay, when you're done talking, please silence your mic, please. The information can flow between a parent cell to daughter cells through DNA replication. That cell will then divide, and then both daughter cells will have a copy of the parent cell's chromosome. That's the flow of information between cells. For humans, you'd say the information flows from two parents to an offspring. And then there's the flow of information between cells of the same generation, where one cell loses DNA, and I've broken this figure to illustrate a little more closely. Usually the parent cell will die, the DNA will come out of it, and it's not a parent cell, it's the donor cell. The DNA will come out of it. The DNA will then be naked DNA in the environment. That naked DNA can be taken up by a recombinant cell, which can then recombine that DNA, and then the cell can gain DNA from another cell. That's the flow of information between cells of the same generation. Any question about this? So when we're talking about the vertical flow of information within a cell, this is the central dogma, that the information flows from DNA to RNA and then to protein. The language of DNA is that there are 4 different nucleotides that this information is stored in. So there are only 4 letters in the alphabet for DNA nucleotides. And then for RNA, the language is also nucleotides. They're a little different. They're not DNA nucleotides, they're RNA nucleotides. And once again, there's only 4 letters in the alphabet, meaning the information is only stored in 4 RNA nucleotides. Now, when the information is translated into protein, the information is stored in the amino acids, and there are 20 different amino acids. So there's 20 letters in the amino acid language. Now for English, for comparison, we have 26 letters in the English language. Any question about any of this? An example of this is the information that's stored in the DNA would be something like that is a dog. When the information is transcribed into RNA, it's going to be similar, but a different dialect. Something like, that is a dog. And when the information is translated into the protein, it'll be quello e un cane, the same information, but in a different language, totally different language. Any question about any of that? All right, let's talk about the flow of information between generations of cells. This happens in DNA replication. DNA replication is semi-conservative, where the 2 strands will separate, and then one strand is used as a template to make a new strand. And then the other strand also is used as a template to make another strand. This is called semi-conservative replication because if we look at the original strand, it would be 2 dark strands of DNA. And then the newly replicated strand will be a light strand of DNA. And then each daughter cell will have 1 new strand and 1 old strand or original strand of DNA, called semi-conservative replication. Conservative replication was the theory that when the DNA replicated, 1 strand would have 2 strands that were both old dark strands of DNA, and then the other cell would have 2 strands of DNA that were both newly light strands. And don't ask me what the liberal model was, because I don't know. Any question about DNA replication? So as I stated, one strand is used as a template in the double strand of DNA to make the newly replicated strand. What happens is the DNA polymerase binds right here on the template strand, sees this is an A here, and will add the corresponding nucleotide, meaning the nucleotide that binds to the A, which is T in this case. And then it will move down a nucleotide on the template, see a C here, and then we'll add a G, which binds to the C. How that adding happens is a tri phosphate nucleoside, meaning a nucleoside triphosphate, gets added, and then two phosphate groups break off. That releases the energy, which is needed to form a covalent bond between this nucleotide, which happens to be the phosphate will bond to the sugar right here. And there we see the diphosphate coming off and the T nucleotide binding with the phosphate binding to the deoxyribose in the previous nucleotide. Any question about that? So this means that one strand of DNA is held together by the sugar phosphate linkage. And that is a covalent chemical bond holding the single strand of DNA together. The double strand of DNA together is held together only by hydrogen bonds that happen between the nucleotide bases, meaning there are two hydrogen bonds that can form between T and A, and three hydrogen bonds that can form between G and C. And it is only these hydrogen bonds that hold the two strands of DNA together. Any question about any of that? All right. If not, let's move on to talking about DNA replication in greater detail. Hopefully that'll load. Where one strand or a double strand is first made into a single strand, two single strands. How that happens is by DNA polymerase. Oh, there it goes. DNA, not polymerase, by DNA helicase. And then one strand of DNA is used as a template, and the DNA polymerase binds to that template and then makes the new strand by adding the correct nucleotide base. Any questions about that?
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right, any questions about that? I think I'll leave that open, and I'll have a shorter time opening the next one. I don't know why we're here. So let's go through the DNA replication in a little greater detail. Guess I can go back. So the DNA strands run in opposite directions, meaning there's a 5' end on one strand over here and a 3' end on the other side over here. And then on this one here, this would be the 3' end, and this would be the 5' end. So when DNA gets replicated, the leading strand is running in the right direction, and the DNA polymerase can bind to the template strand and then move continuously in the correct direction, replicating from 5' to 3'. Now, obviously, you have to have the helicase separating the DNA first, and you have to have the primer put in because you need the RNA primer to get the DNA replication started. Any questions about DNA replication in the leading strand? The other strand, called the lagging strand, cannot be replicated in that manner because the DNA strand is in the wrong orientation. DNA replication has to move from 5' to 3', and that's not the way this strand is set down. So what the DNA replication does is it will make the replication in a short fragment called an Okazaki fragment. And what happens is you'll have a DNA primer start the process, and not a DNA, RNA primer start the process. And then the DNA polymerase binds to the template and the RNA primer, and then fills in the Okazaki fragment, but it can't go very far because immediately above it is another Okazaki fragment. And it's being replicated in the same direction, 5' to 3'. And so in the lagging strand, you have many Okazaki fragments being made to get this strand replicated. Now, why that's important is because on the lagging strand, whenever you put in an Okazaki fragment, there will be two places where the DNA will not be linked together. And that will be the start of the Okazaki fragment and then the end of the Okazaki fragment. Whereas on the leading strand, if the DNA polymerase were to begin at the end of the chromosome here, and if it were to synthesize the DNA continuously and never fall off, you could have the entire chromosome synthesized with one DNA polymerase, and there would be no gap in the DNA, meaning no breaks in the DNA. Now, in reality, the DNA polymerase frequently falls off, and it might not start at the end or beginning of the chromosome, meaning it might start midway through the chromosome. And then this part here would be filled in by another DNA polymerase. And let's say this one right here happens to fall off there. Well, DNA replication doesn't end. What happens is another DNA polymerase will bind right here. Come on. You probably have to have a primer start the process, but I'm going to ignore the primer part for right now. The DNA polymerase binds right here and then continues sequencing the DNA. But right there, where this strand of DNA ended and the new strand began, there will not be a link. There will not be a covalent chemical bond linking that strand of DNA to this strand of DNA. And that's where the enzyme ligase comes in. Wherever there is a break in the link, ligase will join the link together, meaning in this case where I have one strand here, and then that strand continuing there by another polymerase, there'll be a break there. Ligase will bind that together. That means on the leading strand, there will not be very many places where the ligase will need to work. It could even be zero if the DNA polymerase begins at the beginning of the chromosome. Come on, mouse. There it is, at the beginning of the chromosome. And if it never falls off and totally replicates the entire chromosome, ligase would never need to work on the leading strand. But usually it works, I don't know, one or a few times. And that's just because DNA polymerase falls off and the replication doesn't end, but another DNA polymerase binds. And that means ligase has to link the two replicated strands together. Now, on the lagging strand, ligase is working twice at each Okazaki fragment, meaning come on, mouse, at the end of the Okazaki fragment, it'll have to link this Okazaki fragment to that Okazaki fragment. And then at the beginning of the Okazaki fragment, ligase has to link this Okazaki fragment to this Okazaki fragment, which this one is not completed. It'll come all the way up to here. Any question about that? So ligase works quite a bit on the lagging strand. And it's called the lagging strand because the way DNA is replicated in Okazaki fragments, and then this strand lags a little bit from the replication of this strand. And that's just because the replication on this strand is so much more complex. Any questions about any of that? So new DNA is made always in the 5' to 3' direction. The DNA replication is started by an RNA primer. On the leading strand, the DNA is synthesized continuously. On the lagging strand, the DNA is synthesized discontinuously. in several fragments. Let's even call it many fragments. It's more than just a few. So many fragments, and they're called Okazaki fragments after Dr. Okazaki, who discovered it. And then ligase links the different parts of the newly replicated DNA together, which on the lagging strand will be twice for each Okazaki fragment. Any questions about any of that? All right, if not, let's move on to RNA transcription. Transcription is the synthesis of RNA from DNA. DNA is transcribed to make RNA. And there are three types of RNA that are made: messenger RNA, tRNA, or transfer RNA, and messenger RNA is also abbreviated mRNA, and then ribosomal RNA, which is abbreviated rRNA. So when the RNA polymerase is transcribing DNA, how does the RNA polymerase know which RNA to make? This is a question that is actually a very good genetics question. How does the RNA polymerase know which RNA to make? I think we've already covered that there's no difference between messenger RNA, tRNA, and rRNA in the nucleotides. The only difference is the sequence of RNA nucleotides. So the only difference is the sequence of the nucleotides. But how does the RNA polymerase know which RNA to make? From reading the DNA. Any guesses? All right, transcription begins when RNA polymerase binds to the promoter. The promoter is a region of DNA upstream of the gene that is being transcribed. And the function of the promoter is it just tells the RNA polymerase to bind here. And then transcription proceeds in the direction 5 to 3 prime. So it's always 5 to 3 prime, even in RNA transcription as well as in DNA polymerase. And then RNA is made in the 5 to 3 prime direction. The RNA transcription stops when the RNA polymerase reaches the terminator. The terminator is a region of the gene after the gene, and the function of the terminator is it codes for the RNA polymerase to fall off the DNA, and that stops transcription. And nobody ever answered, how does the RNA polymerase know which RNA to make? It's really quite simple. And that is the RNA polymerase is transcribing the DNA. And if the gene codes for a protein, it'll transcribe the RNA as messenger RNA. But if the gene codes for tRNA, then the RNA polymerase will transcribe tRNA. And then if the gene codes for rRNA, the RNA polymerase will transcribe the DNA as rRNA. So the RNA polymerase doesn't need to know which RNA to make because all it is doing is transcribing the DNA, and it's whatever the DNA codes for, which is what the RNA polymerase will make, either messenger RNA, tRNA, or rRNA. Now let me remind you that most genes code for a protein. So most genes will be transcribed as messenger RNA. But there are a few genes that code for tRNA and a few genes that code for rRNA, and they will be transcribed as tRNA and rRNA, respectively. And then to make things further, which you don't need to know this, but just to enlighten you, the majority of the RNA in a cell is actually ribosomal RNA. So most of the genes code for messenger RNA, but the cell makes more rRNA than any other type of RNA. And you don't need to know that last point. I'm just trying to enlighten you. Any question about any of that? All right, here is RNA transcription being shown to you. It's not really an electron microscopic image. This is a fluorescent image, which is using an electron microscope to show the fluorescence. But you can see here is the DNA. There's the RNA polymerase, and there's a stretch of RNA starting to be made. I'm not showing you RNA transcription in an actual cell. Any question about any of that? I think I've already talked about this, but let's go through it in a little more detail. And we have a little video to watch. I think this link doesn't work, so I've got to find another one. Yeah, that's a movie I'll show you later, so. Stop that. How long is that? You want to see how long that is? Let's try that. Don't know. I'll have to skip the DNA replication, but we'll move on to RNA transcription. That's a little too short, I think. We can start that one. We'll just skip the translation part, but let me try this.
From #1 Color Silk, color that actually improves scalp and hair.
Commercial.
New Revlon Tone Gloss Care Hair Color.
DNA replication, let's talk about transcription. The first thing you need to know is that transcription has nothing to do with cell replication processes such as DNA replication, mitosis, or cell division. So, what is transcription, and why is it necessary? Well, transcription is the first step in the process of using the genetic code in DNA to synthesize or build all the different proteins in your body. One problem with synthesizing these proteins is that the instructions for making them are in the DNA, which is located inside the nucleus. But the place proteins are always made is outside the nucleus, either in the ribosomes floating in the cytoplasm or in the ribosomes embedded in the rough endoplasmic reticulum. So, how does the genetic code for synthesizing proteins get from the DNA to the ribosome? DNA uses a messenger called messenger RNA or mRNA to carry the genetic code from the nucleus to the ribosome. The process of building this messenger RNA is called transcription. Now, let's see how transcription happens. Transcription begins when an enzyme called RNA polymerase attaches to a segment of DNA called a gene. A gene contains the code to build a specific protein, which is a macromolecule made up of a sequence of amino acids in a specific order. And within a gene, the specific order of nitrogenous bases dictates the order of amino acids that will make up the protein. Each group of 3 consecutive bases in the gene is actually a cobalt.
We'll get
Each
into that
group is
later.
referred to a particular area of the DNA helix to unwind and separate into two strands. One of the strands, often called the template strand, is the side of DNA that is read or transcribed by the messenger RNA. The other strand of DNA, often called the non-template strand, isn't transcribed by the messenger RNA. So, how are DNA instructions transcribed into messenger RNA? Well, using the template strand as a guide, RNA polymerase uses the base pair rule to assemble free nucleotides in the nucleus into a complementary strand of RNA. For example, RNA polymerase reads the DNA base thymine on the template strand, then binds it to a free nucleotide containing adenine. This process continues with cytosine binding to guanine and guanine binding with cytosine. Remember though, RNA will never contain thymine. So, whenever RNA polymerase sees adenine on the DNA template strand, it pairs adenine with uracil. By using the template strand of DNA as a guide, the genetic code from the non-template strand of DNA has actually been transcribed into messenger RNA. When transcription is complete, the messenger RNA, which is small enough to fit through a nuclear pore, takes the genetic code out of the nucleus to the ribosome, the site of protein synthesis. The process of actually building the protein.
We'll talk about that in a little bit. Any questions about RNA transcription? All right, let's move back. I think I'll close that down.
In order for our bodies to function, we need to supply them.
Close that one down too. Here we go. So RNA transcription begins when first the helicase opens up the double-stranded DNA, making it single-strand DNA, and then the RNA polymerase will bind on the promoter region of the gene. That's a region of the gene upstream of the gene. And then the RNA polymerase will move down the template strand of RNA to synthesize, not RNA, template strand of DNA to synthesize the newly replicated, not replicated, newly transcribed strand of RNA. And you can notice it was shown in the video that initially, when the RNA is put on, it will bind with hydrogen bonds to the DNA. And this is trying to show you right here that as the RNA is put on, newly put on, come on, mouse, sorry, my mouse has died. As the RNA nucleotides are initially put on, they will bind to the DNA, and that's a hydrogen bonding between complementary bases, although one base is DNA and the other base is RNA. Come on, because we're transcribing RNA. And initially, the RNA will bind to the DNA. But as you start transcribing the gene, the RNA will fall off, and then it's not binding to the DNA. And so initially it is binding, but as the RNA puts on new nucleotides, the older nucleotides, meaning the ones first transcribed, fall off the DNA. They're no longer hydrogen binding to the DNA, and that allows the DNA to zip up, meaning where the DNA was made single-stranded by the helicase, the DNA will self-wind, and will start rebinding to itself, meaning the double-stranded will start hydrogen binding to itself. And that way the single-stranded DNA is only a little bit, which is the part being actively transcribed. Any question about that? So when we have the entire gene, let's say it's about here and goes to about here, only the part that's been newly transcribed will be hydrogen bonding to the DNA. The rest of the gene will zip up, meaning that the DNA itself, which was at one point single-stranded, it will rehydrogen bind to itself. Any questions about that? And then here's the big difference between RNA transcription and DNA replication. In DNA replication, all of the genome is replicated. In RNA transcription, only the genes are transcribed. So right here, it looks like this region from here to here is a gene. And then this region right here, and potentially this region right there, is not a gene. This region right here and this region right there will not be transcribed. Only the genes are transcribed. In a eukaryote, that's very important because in a eukaryote, most of the DNA is not genetic information, meaning most of the DNA does not code for a gene. In a prokaryote, especially in bacteria, most of the DNA does code for genetic information, meaning the genes make up most of the DNA. And there's very little non-coding DNA in a bacteria. There is some, but there's very little. And in a eukaryote, most of the DNA does not code for genetic information. And we say most of the DNA is non-coding in a eukaryote. And that means in a eukaryote, most of the DNA is never transcribed because only the genes are transcribed. Any question about any of that? All right. Transcription ends when the terminator reaches the terminator. The terminator codes for the RNA polymerase to fall off the DNA. When it falls off the DNA, it comes apart and the newly synthesized RNA then becomes released, and it can then move into the cytoplasm if it's a eukaryote. If it's a prokaryote, this is happening in the cytoplasm. And then the ribosomes can bind to the messenger RNA and translate it into protein. And that would happen if this gene coded for a messenger RNA. Obviously, if this gene codes for tRNA or rRNA, that's once the polymerase reaches the terminator, that's the end of the gene, meaning that rRNA or tRNA that's been transcribed, that's the end product of that gene. Any question about RNA transcription? All right. So after RNA transcription, the messenger RNA will be read by the ribosomes to translate the messenger RNA into protein. Now, in a eukaryote, that messenger RNA has to leave the nucleus and then enter the cytoplasm and bind to the ribosomes. But in bacteria and archaea, the messenger RNA is made in the cytoplasm. So all that's needed is for the ribosomes to bind to the messenger RNA, and then translation can begin. So translation is a synthesis of proteins by ribosomes using the information in the messenger RNA. Messenger RNA is not translated by one nucleotide. It is translated by groups of 3 nucleotides. And we call these groups of 3 nucleotides in the messenger RNA a codon. A codon, one codon will code for one amino acid. And that's why you need 3 nucleotides to be read on the messenger RNA to make the amino acid. You can't just read one nucleotide. You have to read a group of 3 of them to make the amino acid. Translation will begin on the messenger RNA when the ribosome reaches the start codon, and the start codon is always the codon AUG. So where the RNA nucleotides are A, U, and G, the ribosome will begin protein translation. Why that's important is, let's say that the start codon is here on the messenger RNA. That means everything above or before the start codon will not be translated into protein. Any question about any of that? And then the start codon, besides beginning protein translation, it also codes for the first amino acid, and AUG codes for methionine. So AUG does 2 things. It begins protein translation at the position on the messenger RNA where AUG is, and then it also codes for the first amino acid, which is always methionine. And then the ribosome will move down the messenger RNA until it reaches a stop codon. And there are 3 stop codons. You do not need to know the codons, but here they are here. So the first one is UAA. You don't need to memorize that, but it is a stop codon, and that tells the ribosome, stop protein translation here. And so if the stop codon is in this position in the messenger RNA, everything after that position on the messenger RNA will not be translated into protein. Any question about any of that? All right. So translation follows transcription. And in a sense, you have to have DNA replication before transcription, but that's only when the cell first made the cell. So when a cell first made the cell that's being transcribed. Let's look at translation in the genetic code. Each messenger RNA codon codes for 1 amino acid, but organisms typically contain 20 different amino acids. Like humans, only have 20 different amino acids. And surprisingly, most organisms have either 20 or close to 20 amino acids. And if they have a different amino acid than humans have, they will throw out an amino acid. And that's why most organisms have about 20 amino acids, or 20 different codons coding for 20 different amino acids. But there are 64 possible different codons. So why are there so many more codons than possible amino acids? Well, what happens is there can be more than one codon coding for the same amino acid. So on our table of codons, there's actually only 2. codons that code for only one amino acid. And an easier way to say that is there are only two amino acids that have only one codon. The first is the start codon, AUG, codes for methionine. There's no other codon that codes for methionine. So that one you should probably know because the start codon is important. The other one you don't need to know, but it's the amino acid tryptophan. There's only one codon that codes for tryptophan, and it's UGG. You don't need to know that. All the other amino acids have more than one codon coding for that single amino acid. For example, phenylalanine has two codons coding for the amino acid. It can be UUU or UUC. Both of these codons code for that amino acid. And then serine, the amino acid serine, has four different codons that can code for that one amino acid. And then there's a few amino acids that have six codons coding for that amino acid. And you don't need to know this, but leucine is an example of an amino acid that actually has six codons coding for that one amino acid. This redundancy of the codons coding for more than one amino acid, oops, where did I lose that? The redundancy of more than one codon coding for some of the amino acids, most of the amino acids, is actually known as the degeneracy of the code. Any question about any of that? All right, let's take a look at RNA translation, where messenger RNA is translated into protein.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into three stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters.
All right, let me stop it right here and state that where the the growing polypeptide chain is removed from the tRNA, and then this growing polypeptide chain is added to the newly added amino acid. That enzymatic activity is actually happening by a ribosomal enzyme, or an RNA enzyme, I should say. And that is, it's the ribosome that's doing it, but it is the RNA portion of the ribosome that's doing it. So that's the only enzymatic activity we will discuss in this class, which is an enzyme that is actually a RNA. Any question about that? And that is not discussed in your book, but like I said, it is mentioned in this movie, and the protein translation is discussed in your book, but it just doesn't mention that the enzyme that actually removes the growing polypeptide chain from this tRNA and then binds this growing polypeptide chain to the newly added amino acid, that enzymatic activity is actually an RNA enzyme.
These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Okay, any questions about that? I'll leave it there in case there's a question. All right, let's look at translation in a little more detail. Translation always happens in the cytoplasm, even in a eukaryote, and it begins when the ribosome binds to the messenger RNA and then moves down the messenger RNA. So the first upstream portion of the messenger RNA is not translated into protein. It has to wait until the ribosome reaches the codon AUG, and that tells the ribosome begin protein translation here. And we say that the ribosome reads the messenger RNA, but it's really the tRNA which is reading the messenger RNA. And what happens is the tRNA brings in the correct amino acid, and the tRNA will bind to the codon, and we call that the anticodon, the 3 nucleotides which are complementary to the codon. And the anticodon actually will hydrogen bond to the codon, and that will ensure that the tRNA is bringing in the correct nucleotide, or not nucleotide, amino acid. And so these are the components you need. You need the ribosome, which it's in 2 units. You don't need to know that. You need the messenger RNA, and you need the tRNA bringing in the correct amino acids. The ribosome has 2 positions which are important. There's actually a 3rd position, but it's not important. And the first position is where the first tRNA comes in, and the position of the ribosome actually holds the tRNA in place, bringing in the correct amino acid so that the anticodon can hydrogen bond to the codon, bringing in the correct amino acid. There is a second position in the ribosome which can allow the second tRNA to come in, and it too will hydrogen bond with the anticodon to the codon, ensuring that the correct amino acid is brought in, which codes for this codon. You don't need to know that one, but UA does code for the amino acid leucine. After that, the first amino acid in the first tRNA is removed from the tRNA, and then this amino acid is bound to the newly added amino acid, which is lysine, and that's a peptide bond that forms. And as I stated in the movie, how this is happening is by an RNA enzyme removing the amino acid from this tRNA in the first position, and then binding that amino acid to the amino acid in the second position. That will then allow the first position's tRNA to fall out. Once it falls out, the ribosome will then move down one codon. And as it moves down one codon, the tRNA that used to be in the second position now moves to the first position. And that will allow the tRNA to come in the second position, binding to the third codon, bringing in the correct amino acid. And once again, the RNA enzyme of the ribosome will remove the growing polypeptide chain from this tRNA in the first position and link that growing polypeptide chain to the newly added amino acid in the second position. That will allow this tRNA to fall out, and that will allow the ribosome to move down one more codon, and this tRNA, which used to be in the second position, will now move to the first position, allowing the next tRNA to come in and bind to the codon, bring in the correct amino acid. This will continue this way with the ribosome moving down a codon, one codon at a time, putting in new amino acids in the second position, and then binding the amino acids in the first position to the amino acid in the second position. That will allow the tRNA to fall out, and the ribosome will just move down the messenger RNA, adding on new amino acids as the ribosome moves down until the ribosome reaches the stop codon. Once the stop codon is reached, the ribosome will fall off the messenger RNA. That will release the protein from the tRNA and the ribosome. And then this messenger RNA is free to bind to another ribosome to begin protein translation again. Any question about that? And everything after the stop codon is not translated into protein. Okay, any questions about protein translation? If there's no questions about protein translation, let me talk about a few complications of protein translation. The first complication is shown here on this one there and this one here. And as you can notice, the ribosomes are the big red dots or big red heart-shaped things on the messenger RNA, which is a blue line here. And there you can see the messenger RNA there. And each of these dots is a ribosome. And the first complication is each messenger RNA can have more than one ribosome bound to it. And each of these ribosomes can or are making a growing polypeptide, which will become a protein from that one messenger RNA. So the first complication is you can have more than one ribosome bound to one messenger RNA at the same time. And this happens in both eukaryotes and in prokaryotes. Any question about that first complication? In fact, as you notice here, the ribosome is beginning protein translation before the messenger RNA is even finished being made. This only happens in prokaryotes, where the ribosome can bind to a messenger RNA because the messenger RNA is in the cytoplasm of a prokaryote. This does not happen where the ribosome can bind to the messenger RNA before the messenger RNA is finished being made in a eukaryote, because in a eukaryote, the messenger RNA is made in the nucleus. And then after it's made, the messenger RNA moves into the cytoplasm where protein translation occurs. But once the messenger RNA moves into the cytoplasm, more than one ribosome can bind to one messenger RNA, making multiple proteins from that same messenger RNA. So here we're going to make 1, 2, 3, 4, 5 different proteins from one messenger RNA. And that's the other complication, and that is in bacteria and archaea, you can have protein translation beginning before a messenger RNA or the messenger RNA transcription is finished. And that's a complication you have in prokaryotes. Any question about any of that? And here we're actually seeing both messenger RNA transcription being made. And that's why this messenger RNA is smaller than this messenger RNA. This messenger RNA is either close to being finished or is finished. And that one is just starting. And you can see the dots on it, that there's lots of ribosomes on this messenger RNA translating the messenger RNA into protein in a prokaryote. And here the messenger RNA is just getting started, and already it has 2 ribosomes bound to it, beginning protein translation. So that's an actual electromagnetic picture of that complication. In eukaryotes, we don't have the complication where translation occurs before transcription finishes. And that's because transcription occurs in the nucleus, translation occurs in the cytoplasm. However, in eukaryotes, we have another complication, and that is in the gene of eukaryotes, we have DNA as part of that gene that codes for the protein of a messenger RNA. And we call this DNA exons, the DNA of a gene that actually is translated into the gene product, which in this case would be the amino acids of the protein. But in the gene, we also have regions of DNA inside the gene that are called introns, and they do not code for the protein in a messenger RNA. When the RNA transcript is first made, it will include the regions of the DNA called exons, and the regions of the DNA called introns. The intron regions of this original messenger RNA transcript have to be cut out. The term is called splicing. So the introns are spliced out, and then the exons are spliced together. That means just joined together. And then this finished messenger RNA is allowed to leave the nuclear pore of the nuclear membrane. The original messenger RNA transcript is prevented from leaving the nucleus because these introns prevent the pore from allowing the pore to open and then allow the original transcript to leave. So only the finished messenger RNA transcript is allowed to leave the nucleus, and then it'll go in the cytoplasm and be translated into protein. Any question about that complication? That's a real complication because we've never talked about introns, a region of a gene that does not code for the gene product. And all eukaryotes have introns in their genes. Bacteria do not have introns in their genes. So a bacterial gene only has exons. But a eukaryote has both exons and introns, and the introns have to be removed before that finished messenger RNA can leave the nucleus and then go to the cytoplasm to be translated into protein. Any question about that complication? All right, so we talked about the structure and function of the genetic material. We talked about the flow of genetic information. We talked about DNA replication, RNA transcription, and protein translation. Let's now move on to the regulation of bacterial gene expression. How we're talking about bacterial gene expression is by the operon model. After that, we'll then talk about mutation and genetic transfer and recombination. And then we'll end this chapter on talking a little bit, just one slide, on genes and evolution. Cells want to regulate their components. One way a cell can regulate what the cell makes is by controlling the gene expression. And we're going to look at bacterial gene expression. Eukaryote gene expression is very similar. It's just a little more complicated. So one way to regulate what the cell makes is to control gene expression. There are some genes which are always turned on and they're always expressed at a fixed rate, such as a constitutive enzyme is constantly being made in the cell and it's always made at a fixed rate. But there are other enzymes which are expressed only when they are needed. And these enzymes are repressible and inducible enzymes. These are very similar systems, repressible and inducible. The difference is a repressible one is a gene that is normally turned on, but you can turn it off. An inducible gene is a gene that is normally turned off, but when it's needed, the gene can be turned on. Now, that's one way to regulate gene expression. There are other ways. You can also regulate the bacterial products by pre-transcriptional control. Actually, that's an inducible operon and a repressible operon, which we'll talk about an inducible operon. We're not going to talk about a repressible operon, and you will not be tested on it. You can read about it. There's also epigenetic control, which still regulates the gene by a pre-transcriptional control, so it can control the expression of the gene, but it's a very hairy system, and people have been studying it now for over 50 years, And we still don't have a very good understanding of epigenetic control. And it's good for you that we're not going to talk about it and you're not going to be tested on it. And then there's other ways to control the contents of a cell, and that would be post-transcriptional control. We've talked a little bit about this. You can regulate an enzyme by turning it off, by having something bind to the enzyme, changing its shape, and now the enzyme can no longer function. We're not going to talk about transcriptional control here, and you will not be tested on it. Any question about any of that? All right. So to talk about gene expression, we need to talk about an operon. We're going to talk about bacterial operons. A bacterial operon always contains the gene of interest, and that is called a structural gene. On some bacterial operons, you can have more than one structural gene in one operon. For example, the lac operon, which we'll talk about, has 3 structural genes in one operon. An operon is a super gene containing at least one structural gene and perhaps more structural genes, as well as regions of DNA that control the expression of that structural gene. So normally when we're talking about a gene, we mean a structural gene. But each structural gene has controlling regions around that structural gene that control the expression of that structural gene. And 2 of them are shown here, the promoter and the operator. We've already talked about the promoter. What it does is it codes for the DNA to bind to the RNA polymerase. And so the reason why the RNA polymerase binds to the DNA is because there's a promoter. And then that promoter moves down the DNA, transcribing the gene. And then there's a region not shown here, part of the operon, which is the terminator, and it codes for the RNA polymerase to fall off the gene, ending gene transcription or RNA transcription. And we'll talk about the operon next time. But that's what an operon is, a super gene containing the structural gene of interest, as well as controlling regions around the gene. One example is the lactase enzyme is an enzyme in the lac operon, and that's actually the Z structural gene coding for the lactase enzyme. And that operon contains both the Z structural gene, the Y structural gene, and the A structural gene. We'll talk about those next time. It includes the terminator at the end of the operon, and it includes the promoter at the beginning of the operon and the operator. And these 2 are controlling regions that control the expression of this gene. Any question about that? Let me state that eukaryote operons are similar. One difference is they only have one structural gene in each eukaryote operon. And a bacterial operon may have one structural gene or several structural genes. And we'll talk more about that next time. Any question about anything? If there's no questions here, I'll see you at 6:30 in the lab.
I have a question, Professor.
Go on.
So about the lecture 8A, there wasn't enough questions for everyone to do. I was just wondering what we should do about that.
In that case, try to find a question that you think is answered incorrectly and then answer it. Okay?
Okay.
If no question is answered incorrectly, just start all over again.
Okay. Great.
Okay?
Thank you.
All right.
I had a question about the question to 8A and B. So if I answer 8A, do I need to answer 8B or just 8A?
I have to look at the assignment here.
Because I know, like he said, there's not enough questions. So I did answer 8A, but I wasn't sure if I need to do 8B.
Oh, that's not right. Is
Yeah,
that right? Is that all
yeah.
it is?
That's why I think that's why he asked you about 8A, not having a lot of questions.
Uh-huh. So the last question, I didn't even see 8A. Yeah, you only need to answer one question here.
Okay, and then 8B, the lecture 8B, do I need to answer one of those two? Because there's not a lot of questions in that one as well.
8B,
So if you go to 8B.
a worksheet 8B.
Shh, shh. Rania, I need to help you. Sorry about that.
I've got a lot of questions in 8B.
Oh, maybe I missed. Maybe it was a different one. I don't know. I have to go back again.
I don't think that's the one that we have posted in our modules. There's not that many questions in 8B that we can see.
Yeah, that's why I'm kind of
Let
confused.
me see, maybe I haven't updated that. All right, I'll upload the new 8B and then there will be more questions. Now what was your question again? An 8B?
Like she said in the lecture, this one, it doesn't have a lot of questions, so I think that's why I wanted to know what to do on that.
Well, I'm gonna add more questions.
Okay, sounds good. Thank you.
Okay.
I also had a questions regarding our unknown project. Because I know I sent you one of the tests and you sent me the result back. So
Can
I
you
send an
ask that in the lab? Because I'm trying to get this done.
okay,
I've only got
okay,
a few
sounds good.
minutes,
I
okay?
will.
All right,
Thank you.